All right, so now that we have the transcription unit defined and all the three major pieces associated with it, and understand the orientation of upstream versus downstream and how that nomenclature works, we're going to go ahead and move on to the transcription or the substrates that are needed for transcription to actually take place. So, very similarly to DNA replication, we need nucleoside triphosphates. Okay, so but instead of deoxyribose nucleoside triphosphate, as in DNA replication, we are going to have ribonucleoside triphosphates for transcription. And that's where we first start to see ribonucleic acid actually being the backbone of that triphosphate base. So, again, if you recall back to chapter 10, um, our um, our units or our NTPs or D NTPs have three major components: the triphosphate base, the sugar, and then the actual nucleotide base, so A, G, C, or U in this case. So keep in mind our triphosphate, we've got three phosphate groups hooked together and they all still have negative charges. So mRNA is still negatively charged, similar to DNA. We've got our sugar that is going to have an OH group off the 2' carbon, which is much different than our deoxyribose sugar. And then our base is going to be the same. So the same adenine, guanine, and cytosine, but thiamine is going to be switched out for uracil here. So, as we start to build an mRNA strand off of a template DNA, the nucleotides are always going to be added to the 3' end of the RNA molecule. Very similar to DNA replication. So, the RNA or the messenger RNA strand is going to be synthesized in the 5' to 3' direction, and it's going to be done in a complementary and anti-parallel orientation compared to DNA. So complementary means that the complementary nucleotide is actually being brought in by RNA polymerase, and it's going to be in an anti-parallel fashion so that the non-template strand of DNA, like is shown up here, is not going to actually be um, synthesized into messenger RNA. So, the main steps for transcription are, first of all, initiation of RNA synthesis. In this case, it does not require a primer, so we don't need a primer in the case of messenger RNA synthesis like we do with DNA replication. The second major step is that the new nucleotides are going to be always added to the 3' OH end, and then the DNA is going to unwind at the front of the transcription bubble and rewinds as the messenger RNA has been created or that code has been copied into messenger RNA. The transcription apparatus is going to be in the form of RNA polymerase. So this is very different than a DNA replication because RNA polymerase is going to be the single enzyme that carries out all of the required steps of transcription. Okay, so in DNA replication, we had 8 different enzymes that did the job. Here we have one enzyme that does the job, and that one enzyme is RNA polymerase. So RNA polymerase activity is going to be enhanced or be more efficient when accessory proteins are involved. And an accessory protein comes together to join and leave at different stages of the transcription process. So each accessory protein has a very special function and it will actually enhance or slow down the transcription process. So the transcription is going to require an array of proteins as well. But keep in mind, RNA polymerase is doing the work. It's just more efficient if there's certain proteins that are helping it along. All right, so let's take a more directed look at the mechanism associated with RNA or with transcription, and we can't do that until we look at the apparatus associated with transcription. So in bacterial RNA polymerases, keep in mind one enzyme does it all. So it's going to make RNA, messenger RNA, tRNA, and the snRNAs. So what ends up happening is we have our core RNA polymerase as shown over here on the left-hand side, and that core is going to be made up of 2 beta subunits and 2 alpha subunits. And then for the polymerase to be activated, the sigma factor is going to come in and attach itself to the other 4 subunits to form a holoenzyme. So sigma controls the binding of RNA polymerase to the promoter region of the transcription assembly. And so having that RNA polymerase holozyme actually formed is a key component to starting this transcription process. So the sigma factor is going to be different for different promoter regions. So this could even be within the same genome or the same species. You could have different sigma factors. Okay, but again, that sigma factor is going to be the key component to the activation of RNA polymerase. This is a 3D model of what RNA polymerase actually looks like and how it binds to DNA. So our double helix DNA is going to be this green and blue structure here, and the kind of the confetti looking structure or the ribbon structure, that's going to be the RNA polymerase enzyme. So that enzyme is going to come in and surround DNA and bind to it so that it can begin the transcription process. So in eukaryotic species, this is one of our major differences between bacterial and eukaryotic species is the number of polymerases and their function. So RNA polymerase 1, 2, and 3 are found in all eukaryotes. And RNA polymerase 1 really is responsible for transcribing large ribosomal RNAs. RNA polymerase 2 is really responsible for transcribing pre-mRNAs, a few of the snRNAs, a few of the SNOs, and some of the microRNAs. Polymerase 3 is going to be responsible for transcribing transfer RNAs, small ribosomal RNAs, as well as a few others. And then RNA polymerase 4 and 5 we see exclusively in plants. We don't see them in animals at all. And so these are going to be some siRNAs or sequence interfering RNAs. And then RNA polymerase 5 is going to produce molecules that are RNA in nature and take part in chromatin formation. So similar to DNA polymerase, where we have 5 different ones that we know of in DNA replication, in polymerases, eukaryotic RNA polymerases, we've got 5 different ones that are going to be very specific to certain organisms and transcribe very specific types of ribosomes. All right, so let's talk about the initiation or the mechanism of transcription in bacterial cells. We're going to concentrate in bacterial cells, and then we'll point out some differences in eukaryotic cells at the end of the lecture. First of all, very similarly to RNA replication, transcription happens in 3 main steps: initiation, elongation, and termination. Initiation is where the apparatus is going to assemble on the promoter region and begins the synthesis of RNA. Elongation is where we're going to see RNA polymerase move along the DNA, adding more nucleotides to the 3' end. And termination is where we're going to see a stop of transcription and separation from the DNA. So in initiation, there's a variety of steps that have to take place. So this slide just shows four of them. First of all, the promoter has to be recognized. So again, just to orient you to this figure here, we've got our DNA, double-stranded DNA. Our non-template strand is oriented 5' to 3'. Our template strand is oriented 3' to 5'. The promoter region resides upstream from the start site. Therefore, nucleotides or base pairs that are upstream from the start site get the minus designation. And then the coding region begins at the start site, and the base pairs or nucleotides downstream from the start site are designated by a plus. So the promoter region is really, really important because of this recognition step. So the RNA polymerase has to recognize the promoter system. And in bacterial species and even some eukaryotes, it comes in the form of consensus sequences. So we have a consensus sequence that is conserved across many species at -10 and at -35. So RNA polymerase is really looking for these 2 points to figure out that, okay, 10 base pairs downstream of the -10 consensus sequence, we start the transcription of the messenger RNA strand. The second major step associated with initiation is the formation of the transcription bubble. That's going to be similar to DNA replication. And then we're going to see the creation of the first bonds between the ribosomal NTPs. And then we'll see escape of the transcription apparatus from the promoter region. So it'll leave the promoter region and start downstream past the transcription start site. So promoter recognition, we're going to take a little more of a look at this. So promoter recognition is